Welcome to this teaching video for Human Physiology in Ill Health. My name is Jonathan Downham and I have the website criticalcarepractitioner.co.uk. I'm developing a course aimed at nurse practitioners to teach them basic human physiology, so I thought that whilst I was learning it would be a good opportunity to share that learning with others. This then is part of a series of lectures that will expand over time. I hope you find it useful. If you have any comments, please feel free to add them in the comments below. I also have a podcast series which you can find in iTunes. Just go to iTunes, go to the podcast section and type Critical Care Practitioner into the search bar. If you do listen to them, you can subscribe for free to ensure you get the latest episodes, and it would be great if you could rate them for me. I hope you enjoy this resource. As I learn, then you learn. So let's talk about the cellular lipid bilayer. And this is the area that this is the membrane that surrounds each and every cell in our body. And we need to understand the processes that go across this membrane, because substances need to move across this membrane, and they need to move across, so substances need to move across because of growth, metabolic activities, and this includes the movement of fuel, so glucose, etc., and the use of energy, and also sensory perception. And this is things like signals moving to the brain, then back out to the effectors. So remember, we talked about the reflex loop in an earlier lecture. And some of the problems in the body, some of the problems with the cell walls, are the basis of many of the diseases that we suffer from. So, for example, in some of the cell walls, you can have defective transporters. And an example of this is cystic fibrosis. And in cystic fibrosis, chloride is not moving correctly, so water is, as a consequence, not moving correctly either, because these two ions will often follow each other, and the problem that creates is that mucus secreted by the lung becomes very thick. So in cystic fibrosis is a problem with chloride transport. And therefore water, which then causes mucus becomes thick. And I'm sure, as many of you know, that this causes problems for cystic fibrosis sufferers with things like recurrent chest infections, for example. But as well as defective transporters, there could also be defective channels in the cell walls. And we're going to go on to talk about channels in particular in a moment. So this is where there's defect in the movement of things like sodium, potassium and chloride across the membrane. An example of this is something called long QT syndrome, which changes the heart's ability to pump, and this is a problem with defective channels in the walls of the cell membrane. And these cell membranes, if we just move this up a little bit, these cell membranes are often the targets for some of the drug therapies that we use when trying to solve the patient's problems. So these channels, the channels are often the targets of the drugs we use. So an example of this is in hypertension, so with the hypertensive patient. So in hypertension we use diuretics, and diuretics inhibit the ability of a sodium transporter to move sodium across the membrane. So if sodium is inhibited, inhibits can't spell movement of sodium by interfering with the sodium transporter, and as a consequence water can't follow the sodium, so water does not follow, because wherever sodium goes, water tends to follow it as well in a process of osmosis. So water can't follow the sodium, so they are both lost via the urine because the sodium's not reabsorbed, because we're interfering with the channel, then they're both lost via the urine. So the volume of the extracellular fluid is reduced, and therefore the blood pressure falls. So the volume of the extracellular fluid goes down, and as a consequence so does the BP. So that's how diuretics work in hypertension, by inhibiting the movement, or some of the diuretics inhibiting the movement of the sodium transporter. And another quick example is with stomach ulcers. And the use of proton pump inhibitors, which stops the movement of protons across the cell wall. So let's talk a little bit more about the cell membrane. And the cell membrane is made up of something called the phospholipid bilayer. And you can actually just break this word into two different sections. So you've got the phosphate side and you've got the lipid side. So the phosphate, and this is a diagram of an element of the phospholipid bilayer, is made up of, so the phosphate head, which is this part here at the top. So this is the phosphate containing head. And this is said to be polar. So it's charged. It has a charge on it. And it's also said to be hydrophilic, which means that it will mix readily with water. So it mixes with water. And this is important, and we'll see why in a moment. The second part to it is these two tails here, and this is the non-polar, so there is no electrical charge, tail, and the lipids are fatty acids. And these are hydrophobic. which means that they do not mix with water. And this is important, as we'll see in a second. So what happens with the phospholipid bilayer? Well, as we've said, the head is hydrophilic, so it likes water. The tail is hydrophobic, so it doesn't like water. So what tends to happen is that the head will point outwards where there is water, and the tail will head inwards, and it's a bilayer, so you get two parts to it. So we've got a phospholipid here and a phospholipid here, and the hydrophilic tail will point inwards away from the water contained on the outside, whereas the hydrophilic head will point outwards where the water is. So as a consequence, you get this bilayer effect here. And you can see from this diagram over here that this is what the cell wall will then ultimately look like. So if you can imagine, this is going to be the extracellular fluid on the outside of the cell, and then down here you've got the intracellular fluid on the inside of the cell. So you've got all these hydrophilic heads pointing outwards and pointing directly inwards, and then here you've got the hydrophobic tails. So this is the phospholipid bilayer. So phospholipid bilayer of the cell wall. So to go back to our membrane, we've got the extracellular fluid here and the intracellular fluid on this side, and we're talking about material entering the cell moving down its concentration gradient. If you remember the sugar in coffee analogy, where there's a high concentration of sugar at the bottom of the coffee, then gradually it moves throughout the coffee through its concentration gradient. So that's through diffusion, and diffusion is a random movement of cells across the permeable membrane. So if you imagine on this side we've got a high concentration of whatever solute we're talking about, and on this side we've got a low concentration of the same solute, and what will happen therefore is that the solute will move across the cell membrane down the concentration gradient, but this relies on the substance being soluble in lipids. So it's going to be soluble in the lipids, because remember we've got the polar heads of the phospholipid bilayer, but they've got to cross the membrane, which is full of lipids in between, and they've got to be soluble in lipids. If they're not soluble in lipids, then they're going to have a hard time getting across that membrane. So this cell membrane is going to be, this cell membrane here is going to be permeable to that substance, because it's not necessarily going to be permeable to all substances. An example of one example is that urea is just such a substance. It's soluble in lipids, so it's lipid soluble. So it can cross the membrane down its concentration gradient. So let's talk about some of the characteristics of diffusion, because this will later on help you understand some of the processes that are occurring, certainly in ill health. One of the things I'm thinking of is particularly with respiratory disease, and we'll come to that in the future. But so characteristics. So diffusion moves from high to low concentration. So again, remember the sugar in the coffee cup analogy. It does not require any energy at all. Does not require energy. So we're not expending the very valuable ATP, which we'll talk about in a moment. It continues until equilibrium is reached. So we'll continue until what's on one side of the membrane matches what's on the other side of the membrane. It can occur rapidly over short distances, but it occurs more slowly over long distances, so it's proportional to the distance. So if you can imagine, if you put your sugar in the bottom of a very, very, very long, thin tube, and the sugar went all the way to the bottom, compared to a very short tube, then the diffusion process would take longer in the long, thin tube than it would in the short, thin tube. So it's proportional to distance. It's also related to thickness of the membrane. So related to thickness of the membrane. And as well is related to temperature. So the thicker the membrane, the longer the diffusion process will take, and the lower the temperature. And finally, it's inversely related to the size of the molecule. And the diffusion process, as I said before, is particularly relevant in the respiratory system, because the diffusion of gases across the alveolar wall is very dependent on, for example, things like the thickness of the membrane and also the temperature. But the thickness of the membrane, if the membrane becomes thickened through disease, for example, or because the lungs are becoming filled with fluid, then the diffusion process will slow down, which will make gas exchange even worse than it already is. So one of the ways substances are going to cross the cell membranes, if they're not lipid soluble, is through something called transporters. And transporters are proteins within the cell wall. So you can see below we've got our phospholipid bilayer, and if you can imagine once again that this is the extracellular fluid and this is the intracellular fluid, and this is our protein here. And what happens is that these transporters will change shape to allow the passage of substances. So at the moment this one is open to the ECF, so whichever substance it is that we're concerned with can move into this protein. This will then change shape so that now it's open to the ICF. So the substance involved can then move from the extracellular fluid into the protein, which changes shape and then can move into the intracellular fluid. And a good example of this are the GLUT transporters. GLUT transporters. And these facilitate the movement of glucose across the membrane, and all cells have these. So these facilitate the movement of glucose. This is known as facilitated diffusion. It's still important to note, however, that this is a process which can take place in both directions, so we can go both backwards and forwards with this, so they can change shape either way. But these transporters are specific for a given substance, so they're very specific proteins. And like I say, the GLUT transporters are specific for glucose. And they're also a finite number. In the cell membrane. As well as transporters, we've got something called co-transporters, and what these do is they move more than one solute at any one time. So if I can just explain that a little bit more clearly. So once again, we've got the ECF here, so the extracellular fluid, so the outside of the cell, the intracellular fluid, so the inside of the cell. And an example of this is whereby sodium and glucose will move into the cell together. So you've got sodium and glucose moving into the cell together. So what happens is that both these enter the cell, enter the protein, should I say, or the co-transporter, and this loads of substance in one side of the membrane, which then undergoes this conformational change. So it's going to change shape like this one here, and then that's going to release the sodium and the glucose into the inside of the cell, and it requires both substances to be there, both solutes need to be there. And this process with sodium and glucose occurs particularly in the GI and the GI tract and the renal tubules. So GI tract and renal tubules. And this process is known as a symporter. There is another process whereby the solutes are going to be moving in completely opposite directions. So for example, we've got sodium here and we've got hydrogen ions in here, and the sodium is going to move into the cell and the hydrogen is going to move out of the cell. So there's going to be a conformational change when these two ions enter the protein. It's going to change shape, allowing sodium to come in and hydrogen to go out. And this is known as an antiporter. So they're moving in opposite directions. It binds specific solutes, and both solutes have to be present once again. So you've got a symporter on this side where substances are going in the same direction. You've got an antiporter here where one substance is going in and another substance is coming out. So we've talked about diffusion across the cell membrane. We've talked about transporters in the cell membrane. We also need to talk about some of the channels that are in the cell membrane. And channels are proteins within the cell membrane which, when open, will allow movement of the solutes across the membrane. And an example of a channel is something called an aquaporin. And aquaporins will allow the faster movement of water than would otherwise occur by diffusion. But these channels are often gated. And what that means is that the channel is closed and has a regulated opening of the channel. And when the channel is open, the movement then is by diffusion. So the diffusion process won't take place until that channel is open. And this is where the solute is not lipid soluble, but once the channel opens, then it can go through the channel via the diffusion process. And there are several gating mechanisms to talk about. So the first one is ligand gated channels. And ligand gated channels require a binding of a specific chemical in order to open. So this requires binding of a chemical to open. And an example of this, and something that we'll be looking at later on, is acetylcholine. This has to bind to the nicotinic receptor on the skeletal muscle in order for that channel then to open. Another type of gated channel would be a voltage gated channel. And this requires a specific gradient of an electrical charge to open. So this requires a gradient of electrical charge to open. And there is a charge difference across the plasma membrane, which is due in part to the negatively charged proteins inside the cells. So the inside of the cell is more negative relative to the outside. So when the charge level reaches a specific level, and this happens in neurons and muscle cells, it can open channels, it can open voltage gated channels, and an example of this is in the cardiac muscle. You get voltage gated calcium channels. So those channels won't open until the charge across the cell membrane reaches a specific level, and this helps in the contractility of the heart. So an example, as I say, is calcium voltage gated channels across the cell membrane. And then the final type of gated channel is a mechanical gated channel. And this requires a specific tension to open. And an example of this is in smooth muscle around the blood vessels. It has a particular tonic contraction. So if the walls are stretched, then the channels in the cell walls will open, and this allows calcium to enter, which causes the smooth muscle then to contract. So it very much relies on the tonic contraction of the smooth muscle. That tonic contraction change opens the channels in the plasma membrane, allows calcium to enter, which causes the smooth muscle then to contract. Now, there are occasions when substances needed to be moved against the diffusion gradient, so they need to go from an area of low concentration to an area of high concentration. And this is going to require the use of energy. So the example I've got here, once again, we've got the phospholipid bilayer here. So on the outside is the extracellular fluid, and on the inside is the intracellular fluid. And these are calcium ions here. So there's 3 out here, and there's 1 inside the cell. So this calcium ion needs to move outside the cell into the extracellular fluid, but in order to do so it would be moving against its concentration gradient, because there's more calcium ions outside the cell than there is inside the cell. So like I say, this requires the use of energy. So what happens is that as we move along the process, that this ATP, or adenotriphosphate, will bind to the cell, and as will the bind to the protein pump, which is in the membrane of the cell wall. It will bind to the cell, as will the solute here, and then what will happen is that the ATP, this is ATP, and this is the calcium ion. The ATP will be cleaved by the protein, which will cause the protein to actually change shape, to open up, if you like. So it will be cleaved, and we'll end up with some ADP plus some energy. And that energy will open the protein channel, which means that the calcium can then move outside into the extracellular fluid. So it's going against the diffusion gradient, so it's going from an area of low concentration to an area of high concentration. And if we can talk about where that happens in the body. One of the ones that you'll probably come across most commonly is the sodium potassium pump, which moves both of those against their concentration gradient. And also there are calcium pumps. And these are present in the muscle cells. For example, in the cardiac myocyte, calcium can be pumped out of the cell ready for the next contraction. And we also get it in the GI tract, where this process helps produce acid. So this commonly is in the cardiac myocytes. So this, as you can see, is a more active process requiring the use of energy, moving substances against their concentration gradient. So let's marry some of these things together as far as transcellular transport is concerned. Sometimes products need to move completely across the cell, from one side of the cell to the other. And a good example of this is where you have the GI tract moving substances from the GI tract into the bloodstream so that they can then be moved across to other parts of the body. So on this diagram, let's imagine that this is the lumen of the GI tract, and here we have a blood vessel. And we need to get the substance moved across from the lumen, across the cell, across the epithelium. So this is the epithelium of the gut wall. Across there and into the blood supply. And the side facing the blood vessel would be called the basal side, and this side would be called the apical side. And here the combination of this transporter over here and this one over here is known as secondary active transport. So this represents an ATP-ase transporter. And remember that this needs energy to work, and this can move substances against their concentration gradient. And then across here we've just got a more simple transporter on the other side. So you've had a big meal, and as a consequence your glucose levels have gone up. So this represents the sodium glucose transporter, and it's commonly referred to as the SGLT. And it's a co-transporter and a symporter as well. So what happens is that you get your sodium and your glucose. The glucose is going to move against its concentration gradient, and it's going to take sodium with it. So they're going to both move into the cell itself. So the glucose has entered the cell now and is at high concentration levels, and the glucose will just move against with its diffusion gradient and move out of the cell through simple facilitated diffusion, and then move out of the cell and into the blood vessel. The sodium now has to also be moved out of the cell because the concentration has gone up, but this is now having to move against its concentration gradient, and this is using this ATP-ase pump here. So it's an enzyme which cleaves the ATP, releasing some energy, which means that the sodium can move against its concentration gradient, and it moves via this sodium-potassium pump. So you get 3 sodium will move out of the cell, and as a consequence you'll get 2 potassium moving into the cell. So the sodium's moving against its concentration gradient, so it needs this energy in order to do so. So it's using ATP, which is going to be cleaved, creating energy in order for this pump to be able to move things against their concentration gradient. So in a way, the sugar is getting a free ride on the back of the movement of the sodium. So the co-transporter on one side and then the active pump on the other, moving sodium against its gradient, ensures that the glucose can follow the sodium into the cell and then move out of the cell on the other side of the cell wall. So just as a summary of what we talked about the phospholipid bilayer, and how and why that's structured the way that it is, and then we've talked about various mechanisms of substance or solutes getting across this phospholipid bilayer. So we talked about the simple process of diffusion and some of the characteristics of the diffusion. We've talked about simple transporters, and we talked about co-transporters and symporters and antiporters. We also then went on to talk about some of the channels in the cell wall, and we mentioned ligand-gated, voltage-gated and mechanical-gated channels. We then went on to discuss some of the pumps which are going to pump solutes against their concentration gradient, and the fact that this requires some energy. We talked about transcellular transport, which combines a number of different mechanisms, and this is known as secondary active transport. So those are some of the things we've talked about in this lecture. I hope you've enjoyed it, and I hope you found it useful.